All right, so we'll get started. Thank you for everyone who has joined us today for our Peak Summit sessions. This is our second session that we've done now. Today, Kyle Hoffman, who is our applications manager, is going to go over the improved antibody de novo sequencing and the new features in Peaks AB 3.5. So Kyle, you can kind of take over from here.
Great. So yeah, I'm pleased to announce that we're going to be releasing a new build of our PeaksAB software in the coming weeks. And so today I'll go over how we've improved antibody de novo sequencing in the new build and give an overview of the new features. So there's three main points for this webinar. First, I'll discuss background why we need to sequence antibodies and how we do it from LC-MS data. Then I'll go over the new features in PeaksAB 3.5. And lastly, I'll focus on how we're resolving these common sequencing errors using retention time prediction in the new build. So there are a number of different ways or workflows in order to discover new antibodies. I'm just showing a couple examples here involving immunization, B-cell isolation, or enriching for antibodies from blood serum. There are other ways, including yeast and phage display systems. So on the left side here, you can see after immunization, these cells are isolated, and then a hybridoma cell line is generated by fusing them to a melanoma cell line. After hybridization, individual clones are isolated and then screened for having the antibody with binding affinity of interest and neutralizing the target antigen. So although there's been a lot of success with this approach, it's a very laborious process and can take months in order to complete. So another way of doing this can also, from blood serum, just directly pull down your antibody with binding affinity using by pulling down the antigen. And then after eluting the antibody, we can directly analyze that antibody by LC-MS and retrieve the sequence. And so why do we need to do this? Ultimately, once you've identified the antibody sequence, you can reverse clone and synthesize the gene and then develop a strain or system for expressing that monoclonal antibody and do batch production. So in order to sequence antibodies using LC-MS data, we first start by doing multi-enzyme digestion. So we choose 3 or more proteases in order to digest the antibody and generate overlapping peptides. Next, we'll inject these peptide digests into high-resolution mass spectrometer like an Orbitrap. And then once we have the raw data off the instrument, we'll perform de novo sequencing and peptide sequence assembly using PeaksAB. Within PeaksAB, we also do isoleucine-leucine differentiation. And the last step is validating that we have the correct heavy and light chain sequence by matching to the theoretical mass of the intact mass of each protein. Okay, so a key step, of course, is assembling high quality de novo sequences. So here's an example of the de novo mapping view that we have in PeaksAB. So we typically apply a score cutoff or score threshold where we are only including the higher quality de novo sequence peptides in this assembly. And so software goes and looks for overlapping sequences between peptides and assembles both heavy and light chain from end to C terminus. And so here's what the final result typically looks like. So all these different colored bars represent different enzyme digests, and you can see how the different proteases or digestions result in overlapping peptides that span this variable region. So from any particular project, we're able to achieve fairly deep coverage and peptide mapping to each heavy and light chain sequence. And at the top, we provide fragment ion confidence level, which is color coded. So anything in red is greater than 95% confidence in that amino acid assignment. Blue is between 85 and 95. And then anything that's in black are residues we're not confident in. And you may have to go back and sort of Either look for additional de novo peptides that may support that or run more data and add it to the project. And so here's an example that I've created where you can see there's some black residues and a blue one and a gap in the sequence. And the way we fix this and fill this gap in is first we can look for additional peptides or de novo sequence peptides that were not used in the initial assembly. So we have this manual sequence editing tool, and the way this works is you would select a sequence either just upstream or downstream of that gap. So you select a sequence where we have good coverage and high fragment ion confidence in those residues, and then search for additional sequences containing that sequence. And then PeaksAB will quickly show you which peptides were unused, and it will also show you all the peptides that were used in the assembly. And so this can guide you in making sequence edits, which you can simply do by clicking the residue you want to change and then typing in the text box the sequence that can potentially fill in that gap region or improve the coverage and increase the confidence level. So once you're satisfied with the sequence edits you've made, you can click the green check mark, apply these changes, and the software will undergo another round of sequence assembly and produce another sequence result. And as I mentioned, we perform differentiation of leucine and isoleucine, so two residues that are isobaric. The way we do this is use a three-tier system. So we primarily rely on ETHCD or EAD data. And so basically, after electron transfer dissociation of the side chain, we look for the different mass shift between the W and Z ions. So the difference between W and Z ions of corresponding to 43 Daltons would be a leucine residue. And if the difference is 29 Daltons, that would represent an isoleucine residue. If we don't have sufficient ETHCD data or spectra supporting I or L, we also can look at the enzyme cleavage data. So basically, we look at the number of cleavages induced by chymotrypsin and pepsin, which preferentially cut after leucine. Then lastly, we can look at the percent conservation. So using an internal database, we can look at sort of how frequently leucine or isoleucine occurs at each of these positions. But of course, we can't always rely on conservation, especially in the CDR regions that are highly variable. And PeaksAB has an automated report generator. So by simply clicking one generate report button in the summary page, you can choose which parts of the project will go into an HTML report and be exported. So this includes the coverage statistics, the confidence level in each amino acid. You can select which spectra you'd like to export into the report, the peptide mapping view, brief experimental procedure, the IL statistics table, and then what's new with PeaksAB 3.5 actually is you can also export this intact mass figure showing that we've matched the theoretical mass of the sequence to the measured intact mass. So now I'll go into the overview of the new features in PeaksAB 3.5. So here's a list of the major features we're introducing. The first being supporting timsTOF data. So in addition to supporting Orbitrap and SciEx data, we now support Bruker data. And then IL differentiation is also in the sequence validation workflow. So there's two main workflows in PeaksAB. You can perform protein sequencing to retrieve an unknown sequence, but if you suspect or expect a certain sequence to be present in your sample, you can input that sequence and validate whether it's correct or not. Previous versions, we simply match the spectra to the protein sequence that was put in by the user, but now we actually will differentiate I's and L's in each of the peptides that were sequenced. And so if they differ from the sequence that's protein sequence put in by the user, then they won't map to the appropriate region. Previous versions, we introduced N-link glycan profiling. We've added to this by now having the ability to do O-link glycan profiling. Our intact mass tool is now integrated with our protein sequencing workflow. So if you have intact data, you can load that in addition with the bottom-up data, and PeaksAB will assemble that sequence and attempt to match it to the intact masses that were deconvoluted. We've now merged our glycopeptide results into the peptide mapping view, and I'll go into that in a couple slides. And then, in my opinion, the biggest improvement in PeaksAB is adding in retention time prediction in order to differentiate between isobaric residues and select the correct peptide to use in sequence assembly. So I'll focus in on that major feature in the last part of the talk. So here's just a screenshot from PeaksAB showing that we now support timsTOF data in addition to xenotOF and thermal Orbitrap data. So you can see a peptide here with good fragmentation mapping to the CDR3 region of this antibody. And here's an example showing how to set up the sequence validation workflow that also now supports Leu differentiation. So again, if you suspect a certain sequence or antibody sequence in your sample, you could put it in here, and then PeaksAB will essentially perform a database search and map the de novo sequence peptides to that input. However, now we do Leu differentiation. So if we predict that the sequence has an I rather than an L, then it won't be mapped to the input sequence. Okay, so now we've introduced, we've integrated the intact mass deconvolution tool into our sequencing workflow. So during project setup, after you've uploaded your enzyme digestion data or bottom-up data, we now have this intact tab where you can upload intact data. So here's an example where we've uploaded NIST reduced sample. And then next you would set your mass error tolerances and set the mass range that you want to deconvolute raw data. And then you also have the option to adjust the sliding window that's so the width of the window that's used for deconvoluting across the mass range. And here's what the final result looks like. So it also considers a number of common modifications. So you can see here we've matched the light chain with two disulfide bonds, and here we've matched the heavy chain to this peak with pyroglue modification, C-terminal lysine truncation, and two disulfide bonds. And the bottom window shows which peaks were selected when deconvoluting each of the masses shown at the top. Okay, so we have a glycan profiling tool within PeaksAB. This was introduced in PeaksAB 3.0, where we could do enzyme-based glycan profiling. So you can look at relative abundances of glycans at a particular position from one particular sample. So in this case, trypsin, glycopeptides mapping to position 300. And here's basically how we identify the glycopeptides, and we annotate the glycan fragment ions on each of the MS2 spectra. And in the summary page, we'll also provide this table showing all the glycan moieties that were identified, whether they're N or O linked, and the number of glycopeptides with that glycan at each of the glycosylation sites. And so in the glycopeptide tab, you'll see strictly the glycopeptides that map to the glycan site. So here's an example of those N-linked glycans from the trypsin digest. But what's new in 3.5 is now we can do, glycan profiling of O-link glycans in addition to N-link. So here we have 3 different O-link glycan moieties at threonine position 219. And we can look at the relative abundance of those as well. And so all of the glycopeptides are now merged into the peptide mapping view. And this is beneficial because it shows you the actual coverage across these glycosylation sites. So in previous versions, without mapping the glycopeptides, you'd often see a drop in peptide mapping or coverage through the glycan regions. But now by merging the glycopeptides, you can see we have really good coverage across that glycan site. Okay, and the last main point I want to discuss is retention time prediction and how we're using it to resolve common sequencing errors. So here's a figure that's taken from recent publication in the Journal of Proteome Research, and it's showing mass coincidence rates. So basically, when considering particular mass limit, you can have a number of different amino acid combinations or modifications assigned to the same mass within a spectrum. And so, for example, for proline, it's unique to the mass at roughly 93 Daltons. However, at the mass corresponding to asparagine could also correspond to 2 glycine residues, and so on. So, and this is the case for glutamine, has the same mass as glycine and alanine, and common modifications are also considered here, where deaminated glutamine has the same mass as glutamate. So this can often lead to sequencing errors, and so we needed to come up with a way to differentiate between these. The bottom left panel shows the coincidence rates, and so the y-axis is an indication of the specificity as a function of the number of unique assignments to each peak. So, for example, proline would be one that's unique to a certain mass, versus the number of possible assignments up to each of these mass limits indicated on the right. So the blue line is the 200 Dalton upper mass limit that's shown here at the top. But as you can see, if we go up in the mass limits all the way up to 1 kilodalton, the specificity really drops off. And in addition to that, if you decrease in the precision, so if you sort of increase the error tolerance that you're allowing for each assignment, you also decrease the specificity of the peak assignments. And so these sequence errors arise from sort of 3 different ways. You have the isobarics, for example, I and L, or GG and N, which would have the same mass. But you could also have sequence errors from these rotations or inversions, which are often a result from incomplete fragmentation of the peptide. So without having information on the orientation from fragment ions, we don't know which one is correct. And then the third example are combinations of residues. So 3 or more residues where, again, we don't have fragmentation, could add up to the same mass. And if you've ever visited our booth at conferences, you know that we hand out these mass reference sheets. So if you'd like to look into your spectrum further and kind of manually check these, this is a helpful sheet to have on hand. So it tells you what the possible amino acids or different combinations of amino acids are that would add up to the same mass. And so how to resolve isobarics and sequencing errors. One way might be to check what the conserved sequence is by performing multiple sequence alignment. So at the bottom left here, a glutamine that could correspond to a glycine alanine. If the region is conserved, you can kind of guess that this is a sequencing error and it should be a glutamine rather than glycine alanine. Of course, that will not apply to CDR regions that are highly variable and you can't find sequences in the database. Another way to do this is to manually check the MS2 spectra for maybe a fragment ion or missed assignment of a fragment ion, or maybe there was an assignment to a peak that's down in the background noise level. So shown here, there's a predicted asparagine at the second last position in this peptide, when in fact it should be 2 glycines, and this Y2 ion was missed during sequencing. So this would be a very tedious process if you'd have to go through and manually check each of these peptides. So we needed a better solution for that. So retention time prediction is a way that we can sort of resolve these differences in an automated way. So here's an example. So PeaksAB will perform the first round of de novo sequencing just as it always does. But then we'll generate a database and use this to generate the retention time prediction model. And then once we have the prediction model, we'll do rescoring of the de novo candidates and adjust the ALC scores. So you can see here in the top example, the correct sequence is listed at the top, and it should be a glutamine rather than glycine alanine. And it has the smallest delta RT, which is the difference between your measured and predicted retention time, and also the highest ALC score. And again, another example here. So the second top de novo candidate with glycine alanine containing the sequencing error would have a lower ALC score and a larger delta RT, so it wouldn't be selected for sequence assembly. And so here's another example. Sometimes we have a very similar ALC score. So here the top three have the same ALC score. However, the third one here has the smallest delta RT of 0.29 minutes. And so PeaksAB does a good job in selecting ones after rescoring and preventing these sequence errors from being introduced into the assembly. So in this example, it got the sequence correct and put it in a glutamine residue. And then the last example I'll show here is within a CDR3 of the heavy chain. So again, differentiating between asparagine and two glycines. And you can see we've accurately predicted the retention time. It's only 0.01 minutes different from the measured retention time of the peptide containing two glycines, whereas the peptide with asparagine has a much larger delta RT. So in summary, PeaksAB performs accurate and automated antibody sequencing from LC-MS data. We now support Thermo, SciEx, and Bruker data. Intact mass analysis is integrated with the bottom-up data for sequence validation. And an Olink glycan profiling is included in the new build. And retention time prediction is used to differentiate between de novo candidates with isobaric positions in order to minimize the sequencing errors. So since we have a bit of time left, I can show you some demo projects we've created with Waters monoclonal antibodies. So again, during project setup, after you've added in some of your bottom-up data, you can then click on the intact mass tab, and here is where you would select your intact data, load it in, and then continue to setting your intact mass error and different parameters and the width of the sliding window for deconvolution. The data refinement parameters are the same as previous builds. And then in addition, the protein sequencing project setup part is all the same. We had a question in the earlier session today, so I'll just mention this, is that PeaksAB has the ability to sequence non-antibody proteins as well. So if you have a protein that's different from an antibody and you have some idea of what that protein might be, but you don't know the sequence, you can paste in a reference sequence. So PeaksAB will still perform the de novo sequencing and assembly, but it could use the reference sequence to identify more PSMs in order to improve the coverage and the sequencing result. And then lastly, for glycan profiling, in our previous build, we only had the N-linked, but you can check this box in order to perform the O-link glycan search as well and set your fragment ion tolerance for glycan fragments and the number of different adducts that you'd like to search for. So yeah, those are the new parts of the project setup workflow. And the other thing I'll mention is we have now the intact mass tab. So here is a new tab in the 3.5 build where you can see the assembled light and heavy chains were matched to deconvoluted peaks for both heavy and light chain when considering those modifications. And then the other new part, we have the glycopeptide mapping tab within the protein tab of the result. And all of these glycopeptides are merged into peptide mapping so that you can see good coverage across the glycosylation site. In addition to that, if you'd like to view delta RT values, you can go to the peptide tab. So all the peptides that were used in the assembly of this heavy chain will be listed here. And you can see the measured retention time, predicted retention time for each, and the delta RT values. So yeah, that's basically everything I wanted to present today in this webinar, but of course, I'm happy to take any questions that you might have, and thanks for listening.
Hey, awesome. Thanks so much, Kyle, for presenting that. As Kyle mentioned, if anyone has questions, please feel welcome to either type them in the chat box or unmute yourself and just ask.